Good morning. These are the notes for DNA replication. These should be your second set of notes in this Unit 6. They should be on page 58. You're going to need your Cornell notes set up on the side and your level of organization highlighted correctly, which the level we're doing today is molecule. The title is DNA replication, or S phase. So this is the S phase in the cell cycle that we just got done learning about. Okay, so DNA is like an owner's manual for your body. It has all the instructions and directions for operating and maintaining your body. So everything about you, every single little tiny thing is controlled by your DNA. Okay, so cells are very protective of their copy of the owner's manual, and they refuse to share. So every new cell has to have its own copy of the manual. Every single cell has all of the instructions for everything in your body. It doesn't use all of the instructions, but it has a copy. So if your cells were like a library, every library has its own set of encyclopedias, which I don't even know if y'all know what encyclopedias are. But so you have a set of books, and every library has their own set of books. Maybe when you go to use an encyclopedia, you don't need to use everything. If you're looking up George Washington, you don't need to start in A, you can go right to W for Washington. Okay, but it has the whole set. Okay, in order to give every cell its own set of instructions, a new copy has to be made. And this is what happens in DNA replication. Okay, so replication means to make a copy. And before cells can divide, they have to make their own, they have to make a double copy of DNA so that the new cell has its own set. Okay, there are two steps. Step one, we've got DNA heliocase. It's Helicase. Heliocase ends in ase, so it's an enzyme, and it means it's a protein. Okay, it unzips the DNA strand. So DNA is a double strand, and DNA helicase comes along and it unzips it, so we've got the strands are separated. Okay, and then step two, DNA polymerase, which is an ACE, so it's also an enzyme, which means it's a protein. It matches up the complementary bases for the separate strands to make two new complete strands. So the complementary bases means A and T and C and G. So adenine and thymine, everywhere there's an adenine, a thymine will attach. Everywhere there's a guanine, a cytosine will attach. Okay, when it does that, that makes two complete copies of the DNA that are exactly the same. It's very important that they're exactly the same, otherwise you would end up looking very weird and probably dead. Okay, so this happens during the S phase. S is for synthesis of new DNA of the cell cycle, which happens during interphase. So if you remember, interphase has three phases, G1, S, and G2. Growth, then it does DNA replication, and then it prepares for mitosis. Okay, so these two strands of DNA stay attached at a centromere until they are separated during mitosis. Okay, we just got done learning about mitosis. That is when the two sets of DNA that are made separate, so each new cell has its own complete copy of the DNA. All right, your notes over here on the side. So DNA is the code for life. Everything that's alive uses DNA. Even some things that are not alive, like viruses, technically not alive, but they use DNA. Okay, every cell has the exact same copy. The DNA in your eye cells are exactly the same as the DNA in your fingernail cells. Every single cell has the exact same copy. They don't use every bit of it, but it's there. Okay, and so one way to remember what heliocase does is heliocase unzips your genes. So there's a joke, is if I were an enzyme, I'd want to be DNA heliocase so I could unzip your genes. So funny. So these are the notes for DNA replication on page 58. You will have a foldable that will be done on page 57.